Good morning, I am Ranjan from Goethe University of Frankfurt. Today I will be talking about my work on miRNA, a time series analysis framework for paired microRNA and RNA-seq data that reveals interesting regulatory dynamics. MicroRNA biogenesis has been extensively studied. Recent studies have reported biogenesis factors like DROSA, DISR, and microRNA degradation enzymes or the TDMD factors like TUTs, TENTs to control the mature microRNA expression, that is, the abundance of the 3p and the 5p arms. These factors we would refer to as the processing factors. Moreover, altered processing of the biogenesis factors and degradation enzymes leads to the generation of microRNA byproducts or the modified microRNA forms called isomers. In this study, we focus on the templated isomers. These have either an addition or deletion of a few nucleotides at either ends, that is, either the 3' or the 5' ends of the canonical mature microRNA form. To put together everything, analyze the microRNAs that undergo altered processing or the DP microRNAs and their effects, we build the framework miRMature. miRMature takes time course samples of paired small RNA-seq and total RNA-seq profiles as input. The framework quantifies the DP microRNAs, search for isomers and possible processing factor associations, and finally analyze associated functional pairs that are regulated by DP microRNAs due to the changing arm ratio expression dynamics. To test our framework, we generated paired small RNA-seq and total RNA-seq profiles from AMI stress-induced mice at 6 time points, day 0, day 1, day 3, day 7, day 14 and day 28 from 4 major cell types of the heart: cardiomyocytes, endothelial cells, fibroblasts, and hematopoietic cells. We pooled 3 biological replicates to make 4 technical replicates, followed by small RNA and total RNA sequencing. We found among the small RNA reads that map to ensemble annotated non-coding RNA types, microRNAs have a major contribution. On the left, among the significantly expressed microRNAs, we see waves of expression dynamics along the time course. On the right, we see a change in coverage dynamics among the mature 3p and the 5p arms of one loci near 106p along the time points. These observations gives us a hint that something could be going on with respect to the mature arm dynamics. To quantify the variation in the arms, we define R as the ratio of the 3p and the 5p arm expression. This line plot shows the representation of arm dynamics and the arm ratio for miR106p in cardiomyocytes based on the coverage dynamics we saw in the previous slide. We define the differentially processed microRNAs or DP microRNAs as the microRNAs that show a significant change in arm ratio at a time point and a significant change in its 3p and 5p target expression fold changes at that time point. This is the workflow for computing the differentially processed microRNAs as we defined them earlier. We take small RNA-seq and total RNA-seq from all time points as input. We compute normalized microRNA and gene expression. Using the microRNA arm ratios, we get the microRNAs that show a significant change in arm ratio at any time point day hash compared to day 0 using Mann-Whitney U-test. The confidence set of microRNA targets were obtained using validated target databases and imposing a correlation cutoff of -0.4. We then use KS test to find the significant change in log4 change of the 3p and 5p targets at day hash with respect to day 0. And finally, we associate the significantly changing log4 change in 3p and 5p targets with the changing arm ratio by relating the time points. This is the plot, figure A, for the arm ratio dynamics for miR106p, violet and red being the 3p and 5p arms respectively. At day 14, we see the maximum change in the arm ratio with respect to day 0. And in figure B, we see a significant change in the target fold change of the arms at day 14 as well. We find that the DP microRNAs are cell type specific, with cardiomyocytes having the most unique DP microRNAs. As isomers are the byproducts of altered processing of biogenesis and TDMD cofactors, we sought to investigate how they relate to the arm ratio changes of the DP microRNAs. Isomer ratios for the DP arms was calculated by taking the mean of the isomer abundances of the arm over the mature arm abundance. The arm ratio and the isomer ratio dynamics was then correlated with the expression dynamics of the processing factors to get the significant associations. Here we see, as a result of the workflow, the correlation graph of the differentially expressed cofactors with the isomer and arm ratios of the DP microRNAs in endothelial cells. We find degradation or the TTMD factors like TUTS, DISTRL2 significantly correlated and hence associated with isomer and arm ratios of the DP microRNAs. In order to associate regulatory functions to the 3P and 5P arms of the DP microRNAs, we developed this workflow. We used the confidence set of 3P and 5P target genes as we did in A. We perform function enrichment analysis of these target genes. We then compute the median gene expression profiles of the genes in each of the functional gene sets. We finally compute the correlation between every pair of median gene expression profiles associated with the functions from the 3P and the 5P arms. We report the anti-correlated functional pairs at a certain threshold of negative 0.8 targeted by the 3P and 5P arms of the DP microRNAs. Here is an example of a DP microRNA miR195A from cardiomyocytes. We see in the line plot the 3P and the 5P arm dynamics, and below the median expression distribution of their target genes that are enriched in focal adhesion and microbodic lumen. These are one of the many functional pairs that are co-regulated by the 3P and 5P arms dynamics of the DP microRNA upon induction of amine stress. These are the significant anti-correlated functional pairs that we obtained for miR195A in cardiomyocytes. Many of the pairs could be related to AMI, such as dilated cardiomyopathy or paroxysmal matrix, while many are yet to be investigated. So, to summarize our hypothesis in the AMI context, with altered processing of either the biogenesis factors like DICER, DROSHA, TTMD factors like TUTS and DISTRL2, or with the recruitment of other stress factors, could lead to changes in the microRNA arm ratios and the influx of their isomers. This in turn leads to the dysregulation of the functional pairs regulated by the changing arm ratio dynamics. Thus, miRarmature framework is able to discover interesting microRNA regulatory dynamics. It allows integration of paired small RNA-seq and long RNA-seq profiles from time course and progression studies. We investigate differentially processed microRNAs, associate biogenesis factors and degradation enzymes, and finally predict the functional pairs co-regulated by the DP microRNA arm dynamics. I would like to thank Professor Marcel Schulz for giving me this opportunity to do the research, my group members for helping me with overall discussions and many insightful comments throughout the project, Florian Neukamp for managing the computational facilities and making it all so seamless during project development, Martin Simon and his team with the sequencing and library preparation, Stephanie DeMiller and her group for generating the data and the experiments, and finally iRNA COSI for allowing me this platform to talk about my research. With this, I am happy to take up any questions. Thank you.